Okay, let's get started. Welcome everyone, in person here in Istanbul, as well as online on YouTube and Zoom, for our first ever BioArch or ARC workshop. This is the workshop on hardware acceleration of bioinformatics workloads, and it's the first time we're holding it with the Recomp conference. I guess this is the 23rd year of Recomp, 25th? 27th? Okay, yeah. It's good to have some systems aspects to Recomp after 27 years, let's say, architecture. So the goal of the workshop is really to present and discuss new ideas in accelerating bioinformatics workloads, and specifically with the co-design of hardware and software and new system designs, new computer architectures. So I think of this as system design for bioinformatics, which is a topic that's going to become increasingly important, in my opinion, because definitely software innovations are not keeping up with the amount of data that we're generating in bioinformatics today, and with the amount of computational analyses that we need to do in the entire bioinformatics stack. And I'm going to give a talk about genome analysis as an example, but there are a bunch of other examples in other bioinformatics domains, in my opinion, that look at like proteins, for example, and other types of data, many types of omics in general. And these are all increasing. Proteomics is another example that's increasing in terms of the data. But genomics has, of course, been developed for a long time, and it's really pushing our limits in terms of our devices and other things. So that's the goal of the workshop. And you can see that it's going to be today. We're starting a bit late due to technical issues with the organization. And what we have done is we basically asked people to submit talks, and we've invited a bunch of talks. So we're going to have a bunch of talks. This is also being live streamed on YouTube, and I assume people are following on YouTube. If you have questions on YouTube, feel free to ask us. I think maybe John will monitor them, or anybody can monitor them if they wish. And we'll try to handle them as much as possible. And then I'll go to the talk soon, but we also have a panel on hardware acceleration. This is for the entire Recomp conference on April 17th afternoon. And we have some confirmed speakers. Mohammed, he is here, Damla, and Ryan. We may have one or two more, let's see. That's to be determined. But we're going to discuss hardware acceleration for the broader Recomp community over there. So this workshop is organized by me and John, who's sitting over there, who's my PhD student. You're going to hear him talk later on and introduce some speakers. Okay, so let me cover the program very quickly. This was supposed to be the opening remarks. As you can see, we're already over time. Not too bad, 1 minute. But I was hoping to actually use this time for actually the talk also, but now it's not going to happen. And then I'm going to give a brief introduction to accelerating genome analysis. This is going to mainly foreshadow some thoughts that I have and some of the talks that are going to come, especially from our group. And then you can see there are a bunch of talks from Gagan, Leonid. Some of these are going to be online because people could not travel, and some of these are going to be in person. And then we're going to have a coffee break, and then a bunch of other talks online and in person, and then lunch break. This is just to tell people on YouTube that when we're not live streaming, there's a break going on. Otherwise, we will live stream. And you can see that there are a bunch of talks, academia and industry, and research labs also. Okay, I don't want to go through all of these clearly. How many talks do we have? 16? 17? 16, okay. Yeah, you can see that we have 16 talks. So hopefully it's going to be exciting. A lot of them are focused on genomics, but not all of them. So I think that also kind of gives you a feel of the field, right? Genomics has clearly been pushing forward, and there's a lot more work in architectures and system design for genomics. But maybe we should also be exploring other stuff that is outside genomics within the bigger domain of bioinformatics. Okay, any questions? Okay, then I'll jump to my talk, which is going to be accelerating genome analysis. So for that, I'll stop sharing the screen, and I'll share the PowerPoint. I think everything is still good, right? Yeah. Okay. Okay, we'll try to stick to the time limit, I think, because we have a tight schedule. But maybe we can have a shorter lunch break if needed. Is there lunch provided here? Does anybody know? You don't know? Okay. Oh, no lunch. Okay, so no lunch provided, but there are quite a few places around, so you can have probably better lunch than conference would provide anyway. Yeah, that's usually the case. Conference food is not good. Stuff outside is much better. Okay, so let me give you a quick overview of accelerating genome analysis. This is supposed to be a longer talk, but I'll try to keep it to 30 minutes. This is going to be more of a personal talk because I'm going to tell you our personal journey on how we started on genome analysis and what we're doing. Clearly, there's other stuff that's going on in genome analysis that I cannot cover in 30 minutes. For that, I'll refer you to some papers that we have been writing with people, in some of whom are in this room. Okay, so as I mentioned, I think system design for bioinformatics is a critical problem. It has large implications, societal implications, scientific, medical, personal implications. And this talk is an overview of accelerating a key step in bioinformatics, which is genome sequence analysis. And even within that, I think, in particular, I'll focus a lot on read mapping. Not exclusively, but mostly, because that's the area that we've been focusing on. So you can see that there's bioinformatics, and then genome sequence analysis, and then there's read mapping. We're really focusing on one area for 10 years or so. So imagine what else can we focus on around. And there are many bottlenecks that exist in accessing and manipulating huge amounts of data during genomic data during analysis. And I'm going to cover some recent ideas in accelerating read mapping. And we've started this actually since September 2006 or so. But more recently, we've been doing a lot more work in the area. So hopefully it'll be interesting. So when we started, what did we think about? Basically, we were kind of dreaming. We wanted to design these genome analysis engines that can perform comprehensive genome analysis very, very quickly, like within a minute, let's say. Of course, that's kind of aggressive, let's say, because within 2 weeks was not very good. And then the questions may be sophisticated, or simple things like this. Which of these DNAs does this DNA segment match with? Or what is the likely genetic disposition of this patient to this drug? That's harder. And yeah, there may be other harder questions. I will not go into a lot of questions because we don't have time in this talk, but you can imagine a lot of things. You can have a device that can sequence your genome and quickly tell you a lot of things about your health. Maybe combine that information from other data that you may have about your life, right? So that was our dream, basically. And I think we're getting closer to that dream, but we're not there yet. I think there's a lot more to do in this area. My internet connection is unstable. Is it good? Okay. Okay, so basically, we need really faster and scalable genome analysis because there are many applications. We can understand genetic variations. We can understand species evolution. We can predict different sort of diseases and outbreaks. And we can also understand what's going on in this room, for example. What kind of microbes are we being exposed to, etc. And we can develop personalized medicine solutions that are tailored to the genetic disposition of people, including other data as well. And there are many, many other applications. And I think there's a bright future because we have technologies that can enable this. We can actually generate a lot of genomic data very, very quickly at high throughput. But we need to somehow figure out how to analyze this data. Before I go into more detail, I will mention some of these papers that we have written together with co-authors, some of whom are here, that talk about accelerating genome analysis. This is a relatively easy introduction, I would say, led by Mohammed, who is here, who's going to give 2 talks later on. And Gagan is going to give a talk that covers part of this. So these are some overview readings that talk about accelerating genome analysis. That's from our group. And this is a more recent one that was invited to the special issue of this journal that is actually more comprehensive, I would say. And we have another paper that's in the works that I'm writing with John right now. That's a shorter one, a very short one, that's going to appear in DAC in June. Okay. So I think before I go into more detail, I would say that we have a problem, we have questions to answer. And for this, it's really a system design problem because we have technology, genomic technology at the low level of this, devices that are generating genomic data. And on top of this, we're building algorithms to sort through that data and make decisions. But we cannot ignore what's going on in between, in my opinion. We really need to design these genomic devices so that we can process the data relatively quickly without moving it too much around, because that's going to be a problem. Okay, this is an agenda, but I will not really stick to this full agenda. So let's continue. So I don't think we need to talk about what's the genome made of in this place. Clearly, we have nucleotide bases and This determines a lot of things. Not everything in our lives, clearly, but this determines some of our personality traits, some of our physical traits for sure, and some of our dispositions to different things. But clearly there are environmental effects that also go into it. And this is real, clearly. People have photographed it, and there are lots of stories, but we don't have time to go over it. But in the end, this is what a genome looks like, at least translated into the 4 nucleotide-based sequences. It's basically a huge text. And the length of the text determines on the species, is based on the species. You can see that humans are kind of in the middle and not the most longest genome. And some other species like viruses are much smaller, as you can see. But basically, this also shows a variety of different species in terms of length, but also all of these are actually can be translated into this text. And the question is basically, if you actually sequence all of these, let's pick these 5, how do you determine what is what, for example? Right? That's not going to be easy. So basically, in DNA sequencing, we want to find the complete sequence of ACGTs. These are the nucleotide bases in an organism's DNA or RNA. The challenge is there's no machine today. Unfortunately, the technologies today take long DNA as input, but they actually chop it into pieces. So we cannot get the full sequence. There's no technology that's really good at that today. Maybe in the future it'll change, but we have not found it yet. And all sequencing machines chop DNA into small pieces, different lengths, and identify these relatively small pieces, but they do not tell you how they fit together. Some of them have some implications of how they fit together, but not perfect. So that's why genome sequencing becomes a problem. Basically, you take large DNA molecule and you chop it into pieces with some technology. And these small DNA fragments are called reads, and they need to be identified somehow. And as I said, current sequencing machines provide small randomized fragments of the original DNA sequence called reads. So you need to map them somehow to answer interesting questions, like what is the genetic disposition that you have for some disease. So I think Mohammed also uses this for like untangling yarn balls, right? You have these yarn balls, and you can think of these as different DNAs. And right now you can untangle them nicely, but we don't have a technology to do that. The technology goes through and cuts through all of these, and you somehow need to figure out which piece belongs to which yarn ball. And of course, this is one example question, right? Sometimes you may want to compare different yarn balls to each other in metagenomics, for example. Okay, so these are some choppers, genome sequencers, and we have a lot of interesting ones, as you can see. Some of these are huge, like this. Some of these are very small, like this. There are even smaller ones. And you can put many small ones together to form a data center or farm of sequencers. Basically, there are more, and all produce data with different properties. And these are more examples on the high throughput domain. So because of these technologies, genome sequencing data has been growing because we are able to actually, we have very good technologies from a relative perspective. So we're able to actually reduce the cost of sequencing a genome significantly. You can see in this curve, I'm going to show this again. As a result, we can generate a lot of genomic data today. This is actually a more newer version of that curve. So you can see that this is the cost of a transistor, and this is the cost of genomic data sequenced. You can see that it's actually reducing much faster than the cost of a transistor, which we've been quite successful with. So essentially, genomic technology is actually much stronger, let's say. Sequencing technology is much stronger than our fabrication technologies, which we've developed for longer, I would say. Okay, so I don't want to make more of that, but basically, you can think of the puzzle as you have this reference genome. It gets kind of you have this reference that you have somehow constructed, and when you sequence someone's genome, you get sequence reads. And our task is to somehow piece together this puzzle. Depending on the analysis, you may want perfect piecing or partial piecing, but in the end, you have to piece together something. And in the worst case, you may have to piece together the entire genome. And this is the data generator. This is the chopper. This actually generates data with different characteristics, as we will see soon. It gives you longer reads, relatively longer reads, but it has more error rate compared to some of the bigger choppers we have. And this is a great data generator. This can generate a lot of data at high throughput, low latency. Unfortunately, this device cannot even analyze data. Basically, you have to move the data somewhere to a laptop or to a data center. So you can think of these as devices that are distributed all over the world, and they're generating lots of data, and we're moving all of that data to somewhere to do the processing. So there's a lot of overheads that we're incurring globally in data analysis. So this was, in terms of the read length and the accuracy of the sequencing, because you have sequencing errors when you actually do the sequencing. This is where nanopore technology kind of sits. You have longer reads. And this is where more traditional high throughput sequencing technologies sit. They have very short reads, like 100 or 300 base pairs, but their accuracy is much closer to 100%. Not perfectly 100%, but closer. So you can see that we have a trade-off. And there are other technologies that are more expensive that sit kind of in the middle, but currently they're expensive. And clearly developing technology is good. The question is, can we ever reach very long reads and very high accuracy at low cost? We're not there yet. So because of this, sequencing is actually very good today, if you will, with these caveats. But data analysis is very much bottlenecked. Basically, we need to map what we've sequenced to a reference genome, for example, and then we can do other studies on it, variant calling, and enable scientific discovery and medical advancements. I'm going through this relatively quickly. Clearly, we don't have a lot of time. But if you're interested in read mapping, for example, how do you map the sequenced reads to a reference genome? Even that area is very rich. As you can see, this is a paper that was led by Mohammed, who's here, that talks about 30 years of, maybe for more than 30 years of read mapping, as you can see. There are lots of technologies that have been developed, and there are some commonalities and there are some differences, which we're not going to be able to get into in this talk. So I'll mention one problem in this talk, which is essentially read mapping. So we're going to go a little bit more detail into read mapping and how to accelerate it. Basically, the problem is the sequencer is too good today. It can generate lots of data, but our computational analysis is slow. So we're really bottlenecked by the computational analysis. These are some numbers that you can find, but it really depends on the analysis also in the end. And basically, we have a huge bottleneck in analyzing or mapping the reads into reference genome. And I like this picture actually because we have different technologies that can, we have a special purpose machine for data generation. This is genomic data generation in this case. This is one machine from Illumina, but you can actually have lots of nanopore machines also over here. They're specialized, they're very fast, reasonably accurate, but we're moving that data to very general purpose machines on the right side, maybe far away. That's what this data movement is. And these general purpose machines are not really fit for analyzing the genomic data that's produced over here. So they're not really designed for this purpose. As a result, we have very slow and inefficient processing capability and large amounts of data movement all over the place, all over the world. And I like this picture because unfortunately, this picture is very similar in other domains as well. Video, for example. We generate, if you replace this with a video camera, we can generate lots of video and images all over the world again, and we're doing the same thing basically. We don't have processing capability. If you use web clicks, that's another thing. On the internet, we generate a lot of data, and we're moving it to machines. So we basically have a huge data movement problem. But genomics is, I think, well, video is actually bad, but genomics is pretty bad also. Okay, so I think the problem that we're going to talk a little bit more about is we need to reconstruct the entire genome from many sequenced reads that is coming out of this machine. And can we do that quickly? And can we also do that with minimal data movement? And in the end, can we somehow do everything without moving data here, if that makes sense? So you could make it personalized and private also if that machine belongs to you, for example, as a human. Okay. So basically, that's the problem of read mapping. How do we map many short DNA fragments called reads to a known reference genome with some differences allowed? We have this DNA logically. Physically, it looks like this. We chop it into pieces using the sequencing machine. And we kind of try to figure out where the pieces map in the reference genome. So it's a statistical process in the end. It's not perfect because there are errors when you chop. And there are also variations of this DNA compared to a particular reference genome that was constructed. Okay, and mapping short reads to a reference genome is challenging in the end. And metagenomic analysis makes this worse actually. You have genetic material directly recovered from environmental samples, for example. You may not know what these references are. Now your task is to kind of classify them, map them to a different reference database so that you can figure out what species they may belong to or they may closely relate to, let's say. Clearly, this task is a lot harder, as you can see. Right? In the previous case, we assumed that we know the reference species. Okay. And basically, this is costly. People use dynamic programming algorithms, and there are lots of papers that are written on this topic, which I'll let you find out. A lot of them are actually in the read mapping survey that Mohammed led that I mentioned earlier. But this is a nice paper that talks about the computational cost. And this is a survey that I just mentioned. Okay, so basically, read mappers today, even though they're optimized, they still spend a lot of time in sequence alignment, more than 60% of the time. This number keeps changing, of course, as you optimize the software. And I'm going to talk about some hardware methods for accelerating this. I'm not going to talk about the software methods. So what does it take to accelerate this? This is a picture from our IEEE micro paper that I mentioned earlier. Basically, there are multiple steps in the read mapping pipeline. Indexing the reference genome, for example, is one step. Filtering what is not going to map nicely to the reference genome or different parts of the reference genome is another step. And finally, sequence alignment is another step. And all of these require software algorithms, clearly. And it's important to optimize the software. But all of these can be accelerated through hardware as well, because they, sequence alignment can be accelerated through hardware, FPGAs, etc. So we're going to talk about that. And this paper covers a lot more than what I mentioned, and the later version covers more. Okay, that's the paper. But I should also say that we have a genomics course that Mohammed and Can have been leading in the past few years. If you want more information, there's a lot more. And you can also attend the course online on YouTube. Any questions? Okay, so I'm going to skip some parts that I normally talk about. I'm going to jump into hardware acceleration. Basically, how can we accelerate this in hardware? I'm going to talk about, as I mentioned, our personal journey. This is a work done by Mohammed some years ago. This is his first paper in his PhD thesis, right? Gatekeeper. And the idea was basically to accelerate pre-alignment in DNA read mapping. I will not go into a lot of detail in any of these papers today. I don't think Mohammed is going to talk about this even, right? He's going to talk about newer stuff. But basically, the takeaway is, by using reconfigurable architectures that are common today, FPGAs, you can accelerate filtering. And the filtering is the idea that which reads should not be considered for which locations in the reference genome. Which ones should you not even compare to in the reference genome. And the takeaway is that most of the reads that you generate should not be compared to most locations in the reference genome. That's the takeaway. And you can accelerate it much faster by FPGAs. And again, you can read the paper for more detail. This is the first FPGA-based filter, and it's real, and the results are real. And these are all freely available online, so people have tried this, people have replicated the results. Since this is relatively old, I'm not going to spend more time on this. So the conclusions over here is that you can greatly speed up read mapping. At the time, it was 10x compared to a state-of-the-art mapper. And we also envisioned at the time that this can be integrated with a sequencing device. Basically, it can enable real-time filtering while sequencing, and this sort of hardware acceleration can hopefully pave the way for on-device genomic analysis. Okay, there's more, and I'll let you to the paper. But this is an example of algorithm architecture co-design. You have the algorithm that was developed like using shifted Hamming distance. Mohammed modified it in different ways. And then we map it to the logic device at the bottom. So, of course, some of the layers you don't go through over here. That's the nice part in reconfigurable computing, but that's the idea over here. I will mention that we actually did more work. This is Mohammed's work also. Essentially, different approaches to pre-alignment filtering that uses different algorithms. So I don't want to go into this detail. I want to go into the next one in a little bit more detail, but Mohammed is going to talk about this, right? Mohammed, Shuji. So I'll let you think when he talks. But I will mention the sneaky snake a little bit because this is an example of algorithmic changes that you can find from different domains that can enable much faster analysis. And the idea here is that when you have correct alignment, it's usually you have a sequence of non-overlapping long matches. And this resembles the problem of approximate single net routing problem in VLSI chip layout. Basically, in VLSI chip layout, you have some entrance and you have some exit, if you will, and you want to route some input signal to some output node. And you have to go through, essentially, some repeaters, if you will. And people have developed algorithms to actually minimize the number of repeaters. And the idea in Sneaky Snake, which Mohammed will talk about in more detail, is to reduce the approximate string matching problem to the single net routing problem in VLSI chip layout. And it turns out this is actually very efficient, as you can see. It's 4 orders more of magnitude more accurate than Shuji and Gatekeeper, which I briefly mentioned. And it can greatly accelerate state-of-the-art CPU sequence aligners. Okay, and the numbers Mohammed will go into more detail in his first or second talk? Second talk, I think. Second one, yes. Okay, later Damla will also talk about another algorithm architecture co-designed for a different purpose here, approximate string matching, not filtering, if you will. And here the idea was to actually have a fast and flexible framework that can accelerate multiple steps of genome sequence analysis, not one step. And this is really the first acceleration framework for genome sequence analysis, approximate string matching. And this also uses another algorithm. Basically, I think this points to the importance of looking at different types of algorithms that are friendly to designing in hardware. So if you want to accelerate something in hardware, maybe you should not start with the existing algorithms that are commonly used in software. That's certainly one approach, but maybe there are other much better ways of designing algorithms and hardware together. So Damla has been looking at the BiTAP algorithm that was developed in the 1990s, for example, that uses simple and bitwise operations to perform approximate string matching. And this is very friendly to hardware, except it has some shortcomings, like how to parallelize it and how to perform traceback were not done in those prior works, and Damla added these. And she basically co-designed scalable and memory efficient algorithms with low power, together with low power and area efficient hardware accelerators. Again, she will talk more about this, that's why I won't go into more detail. But basically, you can do there's an accelerator for edit distance calculation over here, then there's an accelerator for traceback, and they communicate with each other in some way. And this is the edit distance calculation accelerator. It's actually a systolic array-based accelerator, and Damla will talk more about this. I like looking at this because some of these accelerators resemble machine learning accelerators also, at least conceptually. So there may be ways of actually taking advantage of some of the existing machine learning accelerators for these different purposes that we may have. So it's good to have these thoughts in mind, I think. So traceback accelerator is actually very specialized for traceback. Again, I'll let Damla to talk about it. As I mentioned, this is going to be an overview of what's some of the talks that are going to be coming. Okay, and the good, we did this work together with Intel actually, so Intel helped a lot in evaluating the area efficiency and performance and energy of the accelerator. And you can see that the amount of additional hardware you need to add to a state-of-the-art Xeon CPU core at the time is not that bad, and power is also not that bad. But you get significant performance improvements on top of this. So there are multiple use cases of Genome. You can use it for pre-alignment filtering, read alignment, as well as edit distance calculation. Again, I will leave the details to Damla, but there are also other use cases potentially that we have not investigated in that work, right? Overlap finding between reads, hash table-based indexing, and also even generic text search, right? Because approximate string matching is actually more general than genomics. It can be applied to text search, and there's more that can be sped up. And the numbers look quite good, as you can see, especially compared to software approaches that are common today, like Minimap 2. You get huge speed ups, huge power consumptions, more than 100% x speed up. And this is kind of expected because we're actually accelerating things with hardware. Okay, what else should I say over here? And yeah. Any questions? So I'll leave this to Damla. Damla is going to give multiple talks. One is going to be on the work that she has done during her PhD. This is part of it. And then she's going to also talk about some of the works that she's been doing at Bionanogenomics after her PhD. Okay, later Joel is going to give a talk on how to make this even better, basically. So he figured out that GenAsm is not good enough, let's say, and overcame GenAsm's limitations with multiple algorithmic changes that are actually accelerated with multiple different types of devices, let's say. Damla, in her later work, looked into sequence-to-graph mapping, which is an area that has not been investigated as much. So in what I've described so far, you get reads and you try to map them to a linear reference genome. But modern genomics is moving towards more graph-based references to represent the variations within a species, for example, because you may not have a single reference genome representing the entire population of humans, for example. Why not have different types of reference genomes encoded together as a graph that also incorporate the variations across human beings, at least the common variations? And that's the idea. Now it turns out the sequence-to-graph mapping enables a lot more quality because it can distinguish between different populations or different variations. But unfortunately, it's a much more difficult problem because computationally now you have to map onto a graph, not onto a sequence. And there have been algorithms for this clearly, which Damla analyzed and showed that they're bottlenecked, they're not scalable. And there's certainly no prior hardware design. So in her second major work, she targeted this hardware design. And she basically designed a specialized, high-performance, scalable, low-cost algorithm hardware co-design that alleviates bottlenecks and sequence-to-graph mapping. And again, I will not go into the details of this, but basically, this is the sequence-to-graph mapping pipeline that Damla will talk about in more detail. There are a bunch of pre-processing steps where you need to index the graph and construct the graph also. And then you need to analyze the graph, which is based on seeding, filtering, chaining, clustering, and the alignment in the end. And this is the hardware that actually essentially does a lot of those things. Well, especially accelerates this part, right? Not the pre-processing steps. But again, I will leave this to Damla to talk about. But you can see that these are very specialized hardware structures. They can be implemented on FPGA. We implemented on ASIC process. And yeah, there are multiple different parts of the pipeline that gets accelerated using these hardware structures. And if you do this, if you go through the pain of all of this, basically you get, again, significant performance and power improvements compared to pure software designs like Graph Aligner or VG in terms of sequence-to-graph mapping. And also in terms of sequence-to-graph alignment, you can actually get significant speed-ups over state-of-the-art software. So we compared the different types of software because different types of software tackle the different use cases that we target over here. And again, also this is a universal accelerator, so we don't just focus on the sequence-to-graph mapping, but we also do sequence-to-sequence alignment. The numbers here are lower because clearly there are a lot of specialized accelerators that have been designed for sequence-to-sequence alignment in the past, including some of our works, but also some of other people's works. Okay, so again, I'll leave this to Damla, but if you cannot wait for Damla, there's another, I don't know what happened here. Well, okay, yeah, somehow I missed this. Yeah, you can, if you cannot wait for Damla, you can watch her video online also for Miska. I'm not going to talk about base calling. Gagan, who is going to talk next after me, is going to talk about base calling, but we don't have time, so I'm going to skip that one. So let me talk about some more emerging technologies. So far, what we've been talking about is, let's say, things that you can build relatively easily today. You have FPGAs, ASICs, GPUs. I think those are interesting, no question about that. But can we do something that fundamentally eliminates the data moment? All of these previous ones are bottlenecked by data moment. I think we need to push the analysis more towards the memory structures, storage structures, and the sensing structures, where genomic data is stored and generated. That way we can reduce the data moment bottleneck. So that's what we've been also working on. This is one of my favorite pictures. Basically, you have this, let's say, very powerful device today. Processing is done only in this chip that has a lot of SRAM, which is memory. The DRAM stores a lot of data. Storage stores a lot of data. Sensors generate a lot of data. Unfortunately, they have no processing capability. So you have to bring all of the data all the way into a small component in this processor chip to process it. This is the data moment bottleneck, basically. And we are seeing this a lot in the genomic domain. And we're going to have talks that discuss this later on. So basically, we would like to accelerate this. And this is a more general approach, but you can actually have genomic data over here. And we want to basically query the genomic data, ask questions to it, like the ones we have discussed earlier, and then get results. So there are many, many things that need to be done here that I'm not going to go into detail about, because this really changes the paradigm. Because now the processor is not processing the data, but memory, or sensors, or storage is processing the data, and there's a lot to do here. But the problem is, if you want to get rid of this data moment, you really need to design algorithms, in general bioinformatics algorithms, that fit processing in memory. In this case, we talk about mapping and filtering, because that's what we've been working on. But there's more. And Gagan is going to talk about this a little bit, I think, on near memory pre-alignment filtering. Gagan and Mohammed worked together to port near memory to Sneaky Snake to near memory, to do pre-alignment filtering in near memory. And basically, we found out that read mapping is heavily bottlenecked by data moment from main memory. And we wanted to perform read mapping near where data resides using specialized logic. So what we did in this work is to redesign the accelerator logic of Sneaky Snake to exploit near memory computation capability on real FPGA boards. So this is actually real. You can do this in real systems, and that's what we did together with IBM. And we showed that you can significantly improve performance and efficiency of Sneaky Snake compared to an IBM Power 9 CPU. And you can see the results are quite large, like 20x improvements, 130x improvements. And these are both performance and energy. Okay, and this is the system where we accelerated. Basically, you offload some computation to this FPGA board that has high bandwidth, low latency access to memory, and you have specialized logic near memory, essentially. So that's what you can do today. You can get significant performance energy improvements, and you can read more papers. Gagan is going to talk more about that. But in the future, I think we have a lot more opportunity. Basically, in the future, if we actually have new devices that can do in-memory computation, we can improve performance even more. Grim filter is one example that I'm not going to talk about. I don't think there's a talk also. This is one of the earliest works in this area, actually. And we wanted to actually take advantage of these emerging devices where you have a logic layer and the memory layers on top so that we can accelerate genomic analysis in the logic layer. In this case, it was pre-alignment filtering. So this requires changing the algorithms, changing the data structures, how you represent the reference genome, for example. And again, I will not talk more about this, but I will just say other true three-dimensional technologies are under development, so there's going to be a lot more in this area, in my opinion. Let me briefly talk about in-storage filtering. So memory is big, but a lot of the data that we are dealing with in genomics is not fitting in memory. You try hard to make it fit in memory, you go through a lot of hoops, but it's really a storage problem, in my opinion, going into the future. So we really need to think about storage as well. So this is one work Nika is going to talk about later. And the idea is basically, instead of moving data all the way from the storage system to memory and computation units, why don't we actually eliminate that data moment? So I'm going to go through these relatively quickly because I know that we don't have a lot of time. But basically, we've done a lot of work to, overall, as a community, to accelerate computation. So computation overhead has reduced, but unfortunately, data moment overhead is not reduced. We still bring data all the way from the storage system into the computation unit and back and forth. So the idea is, in this work, is to filter reads judiciously in the storage system so that you don't move them. You move only what's necessary, let's say, to main memory caches and the computation unit. So it looks like this pictorially. You move very small amount of data. So what do you filter? Again, I think I'll let Nika talk about that. This is actually important, figuring out how to do this relatively efficiently. And I'll let Nika talk about that. And I'm going to skip that slide. So basically, there are challenges. The challenge is how do you actually cater for many different types of workloads where you can actually filter different amounts of data? And how do you actually design the storage system such that you take advantage of limited hardware resources? And the results that we have are significant. Depending on the application, we get different types of speed up, but you can see that the speed up can be more than 30x, and the energy reduction can be close to 30x over here. So I think these are very promising results because you improve both performance and energy at the same time by 30x. Okay, so Nika will talk more about that. So there's a lot more here that I'm not going to talk about. We keep updating this paper on PIM review that Juan has been contributing a lot to also. And we have other papers also that we've been writing on processing in memory. And if you're interested in processing in memory in general, there's a lot more to do in this area, and you can follow our PIM course that's led by Juan over here. So let me quickly talk about what's happening in industry today in terms of processing in memory. And today we're actually in a good spot that people are actually designing these processing near memory engines. So this is the UpMem company that has processors inside the DRAM chip. Samsung, SK Hynix, Samsung and Meta and Alibaba, they all have processors inside the DRAM chip or on the DRAM module. So basically, people are actually trying to test this concept of putting computation very close to the memory banks. And storage is going to be a lot more, in my opinion. So this is the UpMem processing in DRAM engine that has essentially processors and memory next to each other inside the DRAM chip. And these are real. And you can see that we've been testing the system, and Juan's been delivering a lot of lectures. You can see that this is a general purpose processor. So you can offload a lot of computation, and you can watch a lot of presentations from Juan, as well as papers that we have written that summarize the concept and analysis. So I think there's a lot more to do in this sort of system, and we've been actually developing algorithms for this, for sequence alignment, and Izzat is later going to present our work on UpMem. This is going to appear, I think it's already appeared in Bioinformatics, right? Very close to this month. Okay. Later, Hayo is also going to talk about how we minimize the data moment in multiple ways by consolidating base calling and read mapping, and also non-volatile memory-based processing in memory in this architecture. Again, I don't have time to talk about that. But let me quickly talk about future opportunities, and then we're going to conclude. I think there will be newer genome sequencing technologies, even though these are some relatively new sequencing technologies, nanopore sequencing. We don't quite understand, in my opinion. I think we need to do a lot more over here. But there are also new applications like graph analytics-based genomics. References are getting updated, and we need to develop techniques to actually cater for that. And John is going to talk about that. And we've actually been developing tools like Airlift and FastRemap to facilitate fast reference updates. And there's also new frontiers that Jan is going to deliver a talk, I think, at Recomp. Is that correct? ISMB. Oh, you're not going to talk about the Recomp? Okay.
Blend.
Okay, Blend. Okay. I'm not talking about Blend. Basically, what's happening is we have a lot of raw signals also. So maybe read mapping is not that interesting anymore. Or maybe in some cases it's not interesting. Can we actually directly operate on the raw signal data that's generated by this device and other devices? And that's the idea. So again, this is a teaser. We don't have time to talk about this. And Banu is going to talk about target call, which actually accelerates some of these analysis. Okay, so there's a lot more to do over here, but I'm going to conclude because we don't have time. I will conclude, before I fully conclude, I will say that there are things that are happening in industry. So Illumina has been incorporating FPGAs in their platforms so that they can accelerate analysis. NVIDIA has also been using GPUs in their platforms to accelerate analysis. We can say that, I mean, these are relatively simple things to incorporate, but the good thing is that people are incorporating them. And then NVIDIA has been adding more specialized instructions for dynamic programming, which could be used for other things. And Damla is later going to talk about the work that they've been doing with NVIDIA to accelerate genomics. I think these are definitely interesting, but clearly industry is going to be like shorter term oriented. I think the question is what can we do in the longer term that can really make a huge difference. Okay, so recall our dream. Where are we in our dream? We wanted to have an embedded device that can perform comprehensive genome analysis in real time. We still have a long ways to go, basically. Because data is increasing and the things that we want to do in genome analysis is also becoming more sophisticated. We're kind of greedy. It's a moving target, basically. Our goal has been changing. And we have still a long way to go in terms of energy efficiency, performance, security, and privacy. And we have a huge memory bottleneck that we need to tackle. So let me conclude. I think, hopefully, I've convinced you that system design for bioinformatics is a critical problem. And there will be many talks to convince you if you're not convinced yet. And we've talked about especially read mapping in this talk with some deviations. And hopefully, you've seen that there are a lot of potential improvements that you can get. And there are many opportunities that already exist. And there will be even more opportunities going into the future, especially with new sequencing technologies that we need to really understand and analyze, and also with new applications and use cases that I mentioned toward the end. So basically, we have a bright future for genome analysis and hardware-software co-design to accelerate it. And again, this was a short version, so if you want a longer version of this talk, there are multiple versions. And as I said, we have the genomics course, PIM course, and also an SSD course that can help you get into this topic. This is where I'll stop. Any questions? We're not too bad on time, given that we started 17 minutes late. Any questions on YouTube, Zoom, or?
There's no question.
Okay. Then we should probably move fast. People can always ask questions over email or in some other way. We'll be here the entire day. Who's next? Gagan, okay.